Welcome students. This is for the 5th semester B Pharmacy student, Unit 1, Lecture 2, which last class we have discussed about the cyclimic acid pathway up to cyclimic acid formation and the chorismic acid. Up to cyclimic acid, students have to write and listen to it and see the enzymes responsible for the conversion of the biosynthetic pathway to achieve aromatic compounds in plants. This is lecture 2 continuation. To know about this aromatic amino acids, how it is extended from the cyclimic acid pathway. They used to ask questions. If it is an essay question, students have to write the structure clearly with the enzymes responsible for the conversion of the structural form of the each amino acid. This is actually molecular structure of phenylalanine, and the next one is Tyrosine and Tryptophan. Aromatic amino acids. So when we talk about aromatic amino acids, are amino acids that which we are going to discuss have aromatic ring. First one is we should know the chemical structure. Write clearly in your note and study 2 times. And if you write once, it will be memorized in your brain. It's not a very tough structures. So phenylalanine, you see a benzene with CH2, CH, NH2, COOH. A pyruvic acid is attached with a benzene ring. 3 carbon atom with an amino group. So this is phenylalanine. Alanine we know it's also an essential amino acid, aliphatic amino acid, which is attached with a benzene ring. It is phenylalanine. Tyrosine. Just if you have a para position and OH group in phenylalanine, it's tyrosine. 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 You see very clearly it is also only an OH additional. And the next important aromatic amino acid is tryptophan. If you see tryptophan, it has a benzene 6-membered ring structure and a 5-membered ring structure with nitrogen. And you can have CH2, CH, NH2, COOH. So same thing. You have an alanine group attached with this indole structure. We have this basic aromatic ring or aromatic amino acid tryptophan for various secondary metabolites of indole alkaloids and various even secondary metabolites are having the basic ring with tryptophan. So these 3, there are other aromatic amino acid like histidine are there, but still we concentrate in these 3 aromatic amino acid. So once if you know the structure of the compound, it will be easy for you to remember how this final product is formed from the precursors starting in the cyclimic acid pathway. You should remember about in the class how I explained to remember the cyclimic acid pathway from pyruvic acid and a 4-membered erythrose 4-phosphate and having a 7-membered heptulose, and then cyclization gives rise to the cyclimic acid by dehydrocyclimate, cunic acid, and it gives to cyclimic acid. And from cyclimic acid we are getting this chorismic acid. So from chorismic acid only we will get all these structures, and we need the enzymes responsible for this biosynthetic pathway. The starting material, usually we are using this chorismic acid. This is the chorismate or chorismic acid which is formed from the cyclimic acid. We know the cyclimic acid having 3 OH groups, which we know very well. That is the intermediate main precursor for production of various aromatic compounds, aromatic secondary metabolites. So you need to know about the basic pathway for writing, even if they ask the biosynthetic pathway of aromatic amino acids. So these are the enzymes. Already we have seen some of the enzymes in the last class. And this is additional enzyme we are coming across here is isomerase, mutase and transferase. Already we have seen synthetase, hydratase, hydrogenase. These all we have come across in the initial basic metabolic pathway of cyclimic acid, which we studied in the last class. Here, isomerase is an enzyme that catalyzes the structural rearrangement of isomers. Mutase catalyzes the shifting of a functional group from one position to another within the same molecule or the same substrate. Transferase catalyzes transfer of a functional groups like methyl or phosphate from one molecule to another. So this is what you should remember about these enzymes. We will come across these other enzymes also in this pathway. The pathway, when you talk about in a short manner, cyclimic acid starting from the pyruvate and erythrose 4-phosphate. From cyclimic acid, we are getting this chorismic acid. And this chorismic acid, by means of this chorismate mutase variant or an enzyme, it is converted into the intermediate structure of prephenolic acid or prephenate, which will give rise to phenylalanine and tyrosine. The same chorismic acid will be converted into andronic acid and it gives rise to tryptophan. So we will see in step by step and the structures very clearly, and it will be interesting for you to remember and write. If they ask an essay question or short notes, very important to write the chemical structure. Chorismate mutase is an enzyme which we saw in the previous slide, catalyzes the arrangement of chorismic acid or rearrangement of the chorismic acid to prephenolic acid. So we should know about the structure. You see the chorismate, already we know the chorismic acid which is formed from the cyclimic acid. One OH in the 4th position and COOH with one O C double bond CH2 COOH, chorismic acid. This will be converted into prephenate or prephenic acid by means of chorismate mutase enzyme. You can see the shifting from the 3rd position to the top. So there 2 carboxylic acid is present in the same place. One is COOH and another is CH2COOH. So an ester or COOR or you can say a ketone and as well as an acid is present there. So this structure is prephenate or prephenic acid converted from chorismate is an important step for the aromatic amino acid tryptophan, phenylalanine and tyrosine. This reaction is rare biochemical Claisen rearrangement. This rearrangement, that is shifting from the third position to the number one position of this CH2COCOO, how the carbon atom, that is 3 carbon atom, is getting shifted from the third position to the top position by means of the mutase enzyme. This reaction is called the Claisen rearrangement, where the enzyme focuses the substrate, focuses the chorismate into a specific shape, dramatically speeding up the conversion. You can see the conversion of the step. So once you write, then you will know the structure, how it is made, and you will remember the chorismate mutase. And this overall structure again I am showing you detailedly. You see, this is the chorismate and this is the prephenate or chorismic acid or prephenic acid. We have put in an ionic form, that is why we are telling chorismate, because it can be formed as a salt with another one Cl or Br or any halogenic atom. So chorismic acid or prephenic acid, as we have told, H if you put it become acid in the name. So chorismate, you see 1, 2, 3, 4, 5, 6 in the aromatic structure, which is attached with the 7th O and 8th carbon atom and 9th carbon atom COOH, and shifting from the third position directly to the first position. See the 9th carbon atom CH2, C double bond O, 8th carbon atom and COOH. This is the shifting take place. The chorismate is converted into prephenate, or chorismic acid is converted into prephenic acid. So this is the step which we have to say, this reaction, the Claisen rearrangement, very rare rearrangement, Claisen rearrangement, which takes place in biosynthetic pathways. So you see here, now we are coming to the phenylalanine biosynthesis from this prephenate. Here prephenate is converted into phenyl pyravate in the first step. So prephenate, we know that it is prephenic acid from chorismic acid, number 1 carbon atom containing the CH2, C double bond O, COOH and COOH in this carbon atom number 1 and OH. This will be converted into phenyl pyravate, an aromatic compound, phenyl with a pyruvic acid in the number 1 position. So you see, phenyl pyravate is obtained from prephenate. So prephenate dehydratase is the enzyme. Dehydratase. So prephenate dehydratase catalyzed this reaction. This enzyme removes a carbon or carbonyl group, carbon dioxide, or you can say carbonyl group, and a water molecule from the prephenate to create a precursor, phenyl pyravate, which is responsible to produce the phenylalanine. So this is the phenylalanine structure. You see the complete reaction. First, prephenate is formed from chorismic acid, and it is converted into phenyl pyravate by the enzyme prephenate dehydratase. by removing carbon dioxide and water. And this phenyl pyravate is aminated. This C double bond O, it needs an amino group to give phenylalanine. So you are using an amino transferase enzyme. So a dehydratase enzyme is needed as well as a transferase enzyme is needed. Chorismate mutase enzyme convert into prephenate, and prephenate converts this prephenate to phenyl pyravate by means of a prephenate dehydratase enzyme, and then you are giving glutamate, an amino acid, aliphatic amino acid in naturally, which donates the nitrogen, NH2 amino group, that is amino transferase will take place from glucomate, and it will get converted into alpha ketoglutarate, and your phenyl pyravate will become phenylalanine. So this is the reaction which is more important. Prephenate is converted into phenyl pyravate, and an amino transferase will show this point how a glutamate gives the contribution of nitrogen to the phenylalanine. So phenylalanine is usually present in linseed, milk, wheat. It's present in most of the vegetables, even in beans. Specifically, we are giving some status here. So next aromatic amino acid is tyrosine, biosynthesis. We know that it is only an OH group is attached. So prephenate is here converted into para-hydroxy phenyl pyravate. Not directly phenyl pyravate like phenylalanine. Here we need a para-hydroxy group. So here phenyl group is getting attached with an hydroxyl group in the para position. So prephenate dehydrogenase catalyzed in this reaction. Here dehydrogenase, that is dehydratase, here dehydrogenase, and as well as NAD and NADH or NADP also is possible, DPH. So nicotinamide adenine dinucleotide phosphate or dinucleotide, which can give rise to 4-hydroxy phenyl pyravate from prephenate or prephenic acid. Here you should have to remember that the enzyme is prephenate dehydrogenase, dehydrogenase. There dehydratase. These all enzymes we have come across in the last class itself. So here dehydrogenase. The prephenate dehydrogenase, PDH, catalyze the oxidative decarboxylation of prephenate to form 4-hydroxy phenyl pyravate, releasing carbon dioxide and reducing a cofactor. Either NAD or NADPH also can be given. So here this NADPH can also have the key step in biosynthesis of aromatic amino acid tyrosine. Here you can get the release of carbon dioxide, and when you come to the whole reaction here, see the prephenate, it is obtained from or it is formed from chorismic acid or chorismate by means of mutase enzyme. And this prephenate is converted into 4-hydroxy phenylpyruvate by means of enzyme dehydrogenase, that is prephenate dehydrogenase, in the presence of NADH or NADPH also. So prephenate to 4-hydroxy phenylpyruvate and conversion and addition of an amino transferase is very important by means of the presence of glutamate. It contributes the NH2 group, replaces the C double bond O and gives the amino transferase, and you will get the structure of tyrosine. So tyrosine usually present in almond, chicken, avocado, bananas, most of the foods it is present. And overall, when you want to see how these two related aromatic amino acid tyrosine and phenylalanine in overall reaction, Chorismate is converted into prephenate by means of Chorismate mutase, and prephenate is converted into phenylpyruvate by means of prephenate dehydratase, and then amino transferase in the presence of glutamate, it will be converted into our product phenylalanine. Second one, the same Chorismate is converted into prephenate by means of same Chorismate mutase, and the presence of NAD and NADH or NADP also possible in release of carbon dioxide or carbonyl groups. Phenyl dehydrogenase can give rise to 4-hydroxy phenylpyruvate, and an amino transferase is needed by means of using glutamate or glutamic acid, which will be converted into alpha ketoglutarate and giving rise to the tyrosine. So these two structures, and the important point is Chorismate have to be converted to prephenate, so that we have discussed the important junction where to get these two structures. So this is important, prephenate, prephenic acid. How the rearrangement is taking place from Chorismate to prephenate or chlorosmic acid to prephenic acid. The student have to write an important point so that you can write these two structures. Now we are coming to the tryptophan biosynthesis, the next aromatic amino acid. Here it has 5 steps. Number 1, Chorismate is directly converted to anthracite acid or anthracite. This reaction catalyzed by anthracite synthetase. Here synthetase is taking place. And second step, anthracite, that anthracite acid is converted into N5-phosphoribosyl. Ribosyl means a carbohydrate ring structure, ring or lactone ring structure of 5-membered ring structure. A ribosyl. So phosphoribosyl anthracite, this reaction is catalyzed by anthracite phosphoribosyl transferase. And number 3 is third step, this N5-phosphoribosyl anthracite, because you have added this ribosyl and anthracite acid. So your next third step you have to remember in your writing, phosphoribosyl anthracite. It is converted into enol, a double bond structure. I will show you. It becomes a carbon-carbon, a double bond in between in the structure, enol structure. That is enol 1-ortho-carboxy phenyl amino 1-deoxy ribulose phosphate. This reaction is catalyzed by phosphoribulose anthralate isomerase enzyme. And number 4, this enol 1-ortho carboxyphenyl amino, phenyl amino 1-deoxy ribulose phosphate 2 indole, our structure is coming near, indole 3 glycerol phosphate. This reaction is catalyzed by indole 3 glycerol phosphate synthetase. And number 5th reaction in this, the final one in tryptophan, how it is formed, indole 3 glycerol phosphate to tryptophan, the final reaction. This reaction is catalyzed by tryptophan synthetase. So we are coming across here synthetase, transferase, isomerase, synthetase you can say, or synthase is the correct, synthase and again synthase. So synthase, transferase, isomerase, synthase and again synthase. So here one by one we will see the chemical structures and formation of tryptophan. In number 1, Chorismate 2 anthralate or anthralic acid, Chorismic acid to anthralic acid. This reaction is catalyzed by anthralate synthetase. Conversion of Chorismic acid or Chorismate from Citric acid pathway to anthralate or anthralic acid using either glutamine or ammonia as an amino donor. You need amino group and elimination of pyruvic acid molecule from the Chorismic acid is needed. So you see the structure, we know the Chorismic acid structure, Chorismate. Here glutamic acid or glutamine and removal of pyruvic acid in the presence of anthralate synthetase, you are getting this anthralic acid. A benzene with an NH2 group and COO group. So this is anthralic acid. Second position, NH2 and COOH. So converting Chorismate from Citric acid pathway into anthralic acid using either glutamine or ammonia to give or to transfer to give the NH2 group in the second position. This is what you got a direct compound anthralate from Chorismate. And in the second step you can see here anthralate is converted into N5 phosphoribosyl anthralate. So we are adding a phosphoribosyl compound, a phosphoribosyl and the catalyst by means of an enzyme anthralate phosphoribosyl transferase. You can see this carbon atom, 1, 2, 3, 4 carbon atom, a 5-membered ring structure as shown cyclized lactone ring. You might have seen that is glucose is 6-membered monosaccharides. This is tetrose. So you can see that is a ribosyl, not ribulose, ribosyl. So ribulose is taken, ribulose pyrophosphate, PRPP. So anthralic acid is added with an ribulose pyrophosphate, that is phosphate ribulose pyrophosphate, PP, PRPP. 5-phospho D-ribulose, ribose 1-diphosphate, PRPP, which gives rise to, in the presence of anthralic acid or anthralate phosphoribulose, that is phosphopyro or phosphopyro you can say, usually phosphoribosyl transferase is the enzyme, which transfers the phosphoribulose or ribosyl group from 5-phospho-D-ribose 1-diphospho-PRPP to anthralate forming N5-phosphopyroribosyl anthralate PRA. Once you write, it will be easy. Third step, this 5-phospho ribosyl anthralate or anthralic acid to enol. So what is that enol? You see, this is the place where the 5-membered ring structure of the ribosyl is attached in the nitrogen of the anthralic acid NH, and this is the 5-phosphoribosyl anthralate is converted into an enol. Enol means there is a double bond. You see here, I put one arrow. There you can see a double bond very clearly. In the NH, C double bond, C, CH, OH, CH, OH, CH and phosphorylated. That is enol 1-ortho carboxy phenyl amino 1-deoxy ribulose phosphate. So you are getting CDRP, the reversible isomerization, the enzyme which is used isomerase, N5-phosphoribosyl anthralate isomerase. The reversible isomerization of N5-phospho-beta-D-ribosyl anthralate PRA to 1,2-carboxyl phenyl amino 1-deoxy D-ribulose 5-phosphate CDRP. This is the third step. And the fourth step, this enol 1-ortho carboxy phenyl amino 1-deoxy ribulose phosphate to indole cyclizes. This open chain will get cyclized. Indole 3-glycerol phosphate. This reaction is catalyzed by indole 3-glycerol phosphate synthetase. So conversion of substrate from 1-ortho carboxy phenyl amino 1-deoxy ribulose 5-phosphate, that is CDRP, into a precursor. It is a precursor to get the tryptophan releasing by carbon dioxide. Here it is an irreversible, a ring closure will takes place. You can see here how the open chain of enol, double bond is formed in the previous step, and you got that enol 1-ortho carboxy phenyl amino 1-deoxy ribulose phosphate cyclizes by means of releasing water and carbon dioxide in the presence of indole 3-glycerol phosphate synthetase. You are getting that indole structure. You see that a 6-membered ring attached with a 5-membered cyclized nitrogen NH, and you are getting the side chain, that is CHOH, CHOH, CH2 phosphate, that is glycerol. So indole structure means 6-membered, a benzene with a 5-membered with NH group, that is indole structure with a 1 bond. So when you get an attachment of glycerol, you can say that indole 3-glycerol phosphate is the structure which is obtained in the fourth step. And the final step, indole 3-glycerol phosphate to tryptophan. Very easy. This reaction is catalyzed by tryptophan synthetase. There is 2 points of there. Alpha reaction is possible and beta reaction is possible, as it is having an chiral carbon atom. So you can have alpha reaction or beta reactions of indole D-glyceraldehyde, and as well as it is called reto-aldol cleavage, or you can say PLP dependent condensation. The PLP means a beta reaction requires a pyridoxal 5-phosphate, PLP, pyridoxal 5-phosphate as a cofactor for this reaction. So you can see that indole 3 glycerol phosphate cyclizes and give tryptophan as shown in the picture. And here serine or glyceraldehyde 3 phosphate in the presence of PLP as a cofactor. Pyridoxal 5 phosphate is a cofactor. Releasing of water in the serine gives the nitrogen, and so that the replacement of that OH group you will get a nitrogen NH3, so that tryptophan contains 2 nitrogens as shown in the picture. So you will get the structure. Overall pathway, when you want to see this reaction, you can see here the number 1, Chorismate is converted into anthranilate by means of anthranilate synthetase, and this anthranilate is converted into N5-phosphoribosyl anthranilate by addition of a phosphoribosyl pyrophosphate or PRPP, which can give rise to this compound. And this next step, the third step, this N5-phosphoribosyl anthranilate is converted into enol, 1-ortho-carboxy-phenyl amino 1-deoxyribose phosphate can be obtained. And the next step, the next enzyme is indole 3 glycerol phosphate synthetase. So you are getting the reversible, not irreversible, cyclization of this open chain after indole, after the indole structure, if you want, you will get from enolic structure. And that one is the indole D or indole 3 glycerol phosphate synthetase will give you indole 3 glycerol phosphate as shown. And the fifth step, the final step, you are getting the synthetase enzyme, tryptophan synthetase enzyme, the presence of alpha or beta reaction by serine. It can give rise to the cofactor PLP in the presence of glyceraldehyde 3 phosphate. You will get the tryptophan of the amination of the double bond O will get NH3, and you will get the structure of tryptophan. So this is the overall reaction a student once they write, it's possible. And tryptophan is present in algae, and it is present in fish and chocolates and eggs. Most of the things they are present. An amino acid degradation intermediates possible for writing various essential amino acid, aliphatic amino acids. If they ask, it is possible. Once you write this structure, this one what you studied in the biochemistry of TCA cycle, citric acid cycle. You have to write pyruvic acid can be converted into acetyl-CoA, and it can enter into citrate, and it will convert into isocitrate, alpha-ketoglutarate, and succinate, fumarate, oxaloacetate, and back glucose can also be formed, and acetyl-CoA can also give acetyl-CoA or acetoacetate can also be formed. You see here, this can give an glucogenic or ketogenic, both are purely ketogenic, many compounds, many aliphatic amino acids can be formed. Students have to elaborate it, and you have to find out these and write the structures. It's an assignment, you add it. What you write, please find out. You can get from pyruvic acid alanine, cysteine, glycine, SER means serine, threonine, tryptophan. That is already we have seen, aromatic amino acid. Aliphatic, you see, isoleucine, leucine, lysine, threonine. And acetoacetate can also give leucine, lysine, phenylalanine we have seen, tryptophan we have seen, threonine we have not seen. Then you see alpha-ketoglutarate can give rise to arginine, glutamine, glutamic acid, histidine, proline, and succinyl-CoA can give valine, methionine, isoleucine. Fumarate can give rise to aspartic acid and threonine. And oxaloacetate can give aspartame and aspartic acid. So this is very interesting to know about this, all the aliphatic amino acids in this citric acid, how they formed. It is very simple, at least the student have to know the structure of this aliphatic amino acids. How they are formed, you can write it is formed from the citric acid cycle. Once if you know the citrate, isocitrate, ketoglutarate, succinyl-CoA, fumarate, oxaloacetate, acetyl-CoA, they can give rise to. So write the structure of all this aliphatic amino acid which is mentioned here in the short form. And the reaction of biosynthesis, one step, it's possible for the students to write themselves. And you have to submit to my office. It's very interesting for you to know about these structures. Thank you for listening. Try to listen again, write down the structure by yourself and make more comfortable. A short class I made because it needs some time for the students to listen and write down the structure clearly without losing even any carbon atom or bonds. It's very important. How? It's a very simple, and you should know only 3 aromatic amino acids have discussed today. Thank you. So this is the aromatic cyclimic acid pathway, starting from how the things we have discussed like photosynthesis give rise to the starch. From there you will get the glycolysis and you will get pyruvic acid, and it can give another compound which you can get from various pathways of glycolysis like erythrose 4-phosphate, 4 carbon atom and 3 carbon atom, and cyclysis heptulose which can cyclize and give rise to a cunic acid. Cunic acid is converted into cyclimic acid. Cyclimic acid converted into chorismic acid. Chorismic acid is converted into prephenate acid. Prephenate acid is giving the complete aromatic amino acid, phenylalanine, tyrosine and tryptophan. So make yourself comfortable by means of reading again, writing again. Just you can keep it aside. It's not necessary to read again and again. Only recalling, as I told in the class. Recalling is very important for your examination point of view instead of rereading. Thank you.